All life is based on chemistry. You and I are exquisitely complex chemical beings. But chemistry has to be done in a very carefully controlled environment. And this is why all living things are made of one or more cells, which control the chemical reactions essential to life. My objective in Lecture 47 is to introduce the subject of cells. And this lecture is divided into 3 parts. First, I'm going to review the 200-year process of the discovery of cells and the development of the cell theory. Second, I'm going to look at various kinds of cells and how these are studied by light and electron microscopes. Finally, I'm going to tell you about some of the complex internal structures that we find in all cells. I want to begin with the historical development of this cell theory. There's a central paradigm in physics and chemistry called reductionism. Many physicists, many chemists believe that at root, to understand nature, you have to understand the smallest building block, the smallest unit. And then if you understand that unit and how it interacts with itself, how it puts together larger structures, then you can build up understanding from the smallest scale to the largest. We see this in the study of the atom. We see this in the particle physicists' study of quarks and leptons. And as you will see later on, we see it in the biologist's investigation of genes. But of course, seeing matter at the smallest scale is only half the story, because we live in a macroscopic world. We live in a world of great complexity. If we're interested in whole living organisms, in ecosystems, in the field of consciousness, you can't just resort to the study of atoms and quarks and leptons. For this reason, biologists for a long time resisted any sort of reductionist approach. Organisms and their interactions cannot be understood without some reference to a macroscopic world. It seems almost inconceivable that the complex behavior and strategies of living things can be reduced just to a discussion of a few simple subunits. With the introduction of more and more powerful microscopes, however, reductionism has also contributed to biological thinking, and it has become a central aspect of the cell theory. The first step in this process was the discovery in the 19th century that all organisms are made of cells, cells which are the chemical factories of life. The discovery of cells themselves was made much earlier than that. It was made by the British physicist Robert Hooke, who lived from 1635 to 1702. Hooke was renowned for his ingenious mechanical inventions and his demonstrations of various principles of mechanics. Let me tell you a little bit about Hooke. He's a fascinating character in scientific history. He was born in the British Isle of Wight in 1635. His father was a minister, and Hooke, as a consequence, received an excellent education. He was an outstanding student, and he studied at Oxford University. At the age of 20, he became a lab assistant to the very famous British scientist Robert Boyle, who made many major discoveries in terms of the properties of gases. Hooke helped Boyle by designing the modern air pump, the one that's still used in many laboratories. He devised all sorts of improvements to timepieces using wound springs, tightly coiled springs that hadn't been used in clocks before. He also made very significant contributions to the understanding of light and to the understanding of combustion, fire. Hooke was a central figure in the founding of the Royal Society of London in 1661. That was the first scientific society in the world. And it was Hooke who was assigned the task of performing various demonstrations at the society as the curator of experiments. So when people would get together and meet, Hooke would set up a demonstration and share it with other people to illustrate various physical and chemical principles. Hooke also played a major role with his friend and colleague Christopher Wren in the effort to redesign London after the devastating Great Fire of 1666. Hooke's most famous scientific contribution was a book called Micrographia, published in 1665. In that book, he described and he illustrated in beautiful drawings numerous original observations with the microscope. Indeed, Hooke was really the first scientist to use the microscope systematically, and in this sense, his impact with the microscope was comparable to that of Galileo half a century earlier, who was the first person to use the telescope. The telescope looking at objects in the distance, the microscope looking at objects at the extremely small scale, were in a sense parallel in the way they opened up new worlds of scientific discovery. In micrographia, Hooke observed the microscopic structure of cork. That's a kind of wood structure. And so he coined the word cells because the structure of cork reminded him so much of the cells that housed monks at a monastery. By the way, I should tell you that Hooke's life in science did not in any way lead to a happy or introspective life. He's said to have been a very proud man, a contentious personality. He got into many disputes over scientific priority. He made many enemies, most notably with Isaac Newton. And he reportedly died a bitter old man in 1702, a sad end to a distinguished life. Hooke's naming of cells and his other microscopical studies, along with those of, you may recall, Antony van Leeuwenhoek, who was another microscopist of that period, had very little immediate impact on biology. It was almost a series of curios looking at the microscopic structure. Descriptive botany and descriptive zoology, which involved going out to remote places and describing new species of plants and animals, was still the major effort at the time, and it remained so for the better part of 2 centuries. But the microscope ultimately had a profound effect on biology. In 1838, the German botanist Matthias Schleiden proposed that all plants are made of cells. In the following year, his countryman, zoologist Theodor Schwann, extended this idea to animals, that all animals also were made of cells. And Schwann proposed 3 tenets of what is now known as the cell theory. The first of these tenets is that all living things are composed of cells. That's a profound statement. All living things are composed of cells. This leads to the second tenet, which is the cell is the fundamental building block of life. In a sense, we've come to a kind of reductionism with respect to biology. The third idea is equally profound. All cells arise from previous cells. These ideas initiated the field of cellular biology, and that remains one of the central fields of biology today. Now let's look at cells, which are remarkably varied in their size, in their shape, and even in their function in higher animals and in plants. Most cells, of course, are microscopic. They range in size from only a few 10,000ths of an inch, and that's much too small to see even in normal light microscopes, the kind you'd see in a college laboratory. You need very sophisticated instruments to see these tiniest of cells. Some cells, on the other hand, are many feet long. The nerve cells of a giraffe, extending from the leg up to the brain, can be more than 10 feet long. The largest cell alive today is the yolk of a fertilized ostrich egg. That's a very massive cell, and there are ancient birds, extinct birds, that were even larger than that. Cells share 3 characteristics that are common to all cells. First of all cells are bounded by a cell membrane. Recall we talked about phospholipids, the lipid bilayer. Well, all cells have to be bounded, so there's an inside and an outside, a characteristic common to all cells. A second characteristic is all cells have the ability to survive as isolated units. Well, actually, there's a proviso there. In some animals, there are some cells that have gotten so specialized they can no longer survive. But in general, cells are able to survive as isolated units. Many cells in your body and many cells from plants can be isolated and cultured, and they will survive alone. They'll adopt an amoeba-like shape. They'll actually become individual isolated cells. Third characteristic, which follows from this last idea, is that cells perform all the functions of a living organism. Individual cells have all those characteristics of reproduction and growth, of being able to obtain energy and atoms from their environment, and so forth. It's fascinating that you can actually take individual human cells from various parts of the body and grow them so that they form this amoeba-like organism. You can put them in a nutrient dish. They'll adopt this blob-like shape. They'll multiply and divide just like normal single-celled organisms. And yet they contain all the genetic information required to make a complete human being. We just haven't learned how to clone humans yet from that single cell, but the cells contain all that information, and yet they behave like a single-celled organism. So in a sense, we humans are collective animals. We're collective organisms composed of perhaps 100 trillion cells that are all cooperating, and yet many of those cells are capable of independent existence. Let's look at the different kinds of cells. Biologists recognize two fundamentally different structural types of cells. We have prokaryotes. That's a name meaning before the nucleus. These are cells that are small, simple objects without any well-defined internal structure, such as a nucleus. All prokaryotic cells are bacteria, and they all belong to the kingdom of Monera, at least now. Remember, we may be splitting Monera into two. There may also be archaea, but all archaea are also prokaryotes. They're often extremophiles. They're often organisms that live at an extremely high temperature or at high pressure in other extreme environments. The second kind of cell is called eukaryotes, meaning true nucleus. These cells are generally larger. They have a nucleus and other discrete structures inside the cell, discrete structures that are bounded by their own membranes. All plants, all animals, all fungi, and many kinds of protists, single-celled organisms, are made of eukaryotic cells. Biologists now believe that these two kinds of cells represent different evolutionary pathways that diverged very early in the Earth's history. At this point, I have to talk about microscopes. Cells are so small that they can't be studied or usually even seen by the unaided eye. And so this development of a new kind of instrument, the microscope, was fundamental to understanding the properties of cells, and they continue to be so. Improved microscopes, as in so many different branches of science, improved instruments are the key to making discoveries. It's a theme that constantly pervades science. Scientists use two different kinds of microscopes to study cells. The familiar kind that you've probably seen in high school and college biology classes is a light or an optical microscope. Optical microscopes, of course, use glass lenses to bend and focus light that passes through a sample. There's a basic principle of optics. It states that under normal circumstances, you can't resolve features in an object that are much smaller than the wavelength of radiation that you're using to study them, that you're using to image the object. Visible light has wavelengths that average about 20 millionths of an inch, and so the most powerful light microscope can thus magnify about 1,000 times. That's enough to resolve an object that's maybe as small as 10 millionths of an inch. 10 millionths of an inch. And that's a limitation based on the wavelength of light. Of course, most ordinary microscopes don't get above around 100 power, but very, very sophisticated light microscopes with the best optics possible, you just can't get above about 1,000 magnification. So light microscopes can reveal all different kinds of cells, including even those smallest cells if you have a very good microscope, but you can't really see a lot of the smaller structures inside the cells. And this leads us to a second kind of microscope called an electron microscope, which uses a very finely focused beam of electrons that pass through an extremely thin sample, for example, a cell. In this case, electrons are boiled off a filament, they're accelerated through a voltage, and those electrons are focused on a sample with a series of electromagnets that act as lenses. Electrons, recall, have properties of both particles and waves. The typical wavelength of an electron is about 1/1000th of an inch, which gives you a much higher resolution. That means you can get magnifications of 1 million power, and it allows you to observe detailed structures down to even large molecules that exist within cells. So using these microscopes, let's talk about the structure that we've discovered on the insides of cells. When I took high school biology class quite a while ago, I was told that the cell was filled with a mysterious substance called protoplasm. Today, that's not the way we think about the interior of the cell. With advances in electron microscopy, we've demonstrated that cells have extremely complex internal structures, and these internal structures are isolated from their environment by the cell membrane. We see the cell membrane as being formed primarily from a lipid bilayer. And I want you to recall that these are layers that form spontaneously from individual phospholipid molecules that have a water-loving and a water-repellent end. They self-organize themselves into that double layer so that the outsides of the layer see water both on the inside of the cell and the outside of the cell. But the cell membrane can't be a solid surface. It has to be studded with receptors, protein receptors, that provide doorways for food to enter and for waste to be excreted. The membrane thus regulates the exchange of materials from the inside of the cell to the outside of the cell. And the way this happens is fascinating. You can imagine a protein as having a particular shape as a receptor, and it's sitting there waiting on the surface of a cell for the right kind of food to come along. When that food comes along, it fits into the active side of the protein, and the protein then suddenly changes shape, allowing the material to the inside of the cell. As soon as the material is put into the inside of the cell, the protein changes shape again, ready to take in more food. This also provides a vulnerability, because if a virus can look to the outside world like a piece of food, a protein may accidentally trap onto a virus and let the virus inside the cell. Of course, excreting waste is the same process in reverse. Inside a eukaryotic cell, there are numerous separate structures called organelles. Each organelle is itself surrounded by a cell membrane, and each performs a specific function. There are lots of organelles. I'm not going to go through them all, but certainly you've heard of the nucleus, which is the largest of the organelles. The nucleus contains all the cell's genetic information, the blueprint necessary to perform all those chemical operations and the chemical instructions that make the cell function. You also see that cell division is controlled from the nucleus. And as we're going to see in the next lecture, the storage and the transfer of biological information, which is key to the challenge of all cells to reproduce, is also carried out in that nucleus. As you might expect, there are several organelles that play a role in the assembling and the manufacturing of the chemicals necessary for life. For example, distributing proteins and lipids and other cell materials. Some of these machines, the organelles inside a cell, include the endoplasmic reticulum, the Golgi complex. I love these names. They're wonderful names to organelles. We have lysosomes. They're organelles that contain various digestive enzymes and help to break down food molecules. They're like little digestive stomachs inside the cells. Also, lysosomes can be used to attack and kill invading bacteria and other foreign objects because they have these digestive enzymes that can break down the invader. Lysosomes play fascinating roles in the development of embryos. For example, at a very early stage in the embryo of the human, you actually have webbed fingers and webbed toes. Lysosomes break down the very delicate membrane in between the fingers so that we have separated fingers rather than webbed fingers. And in many cases in the development of organisms, lysosomes come into play, killing cells, breaking down structures to open pathways and channels and so forth in the developing organism. It turns out that if you have severe oxygen deprivation, it can cause cells to become too acidic. When that happens, the lysosome walls actually break down, and cells are killed from the inside because those digestive enzymes are released into the cell at large, and it kills the cell quite quickly. In humans, one of the first places this happens is the brain cells, which is why when you're oxygen deprived, when you're suffocated, the brain can be damaged very quickly in that process in just a matter of minutes as the oxygen supply is cut off. Another part of the cell's interior is called the cytoskeleton. It provides a supporting framework. It provides a kind of conveyor belt system that moves materials from one part of the cell to the other. The cytoskeleton employs a whole array of strong protein fibers that connects every part of the cell with every other part of the cell. We'll also see that organelles play a central role in the energy and the metabolism of every cell. For example, mitochondria. These are organelles that are present in every cell. They're miniature power plants of all eukaryotic cells. They convert the energy-rich carbohydrates, such as glucose, the sugar, into compact battery-like ATP molecules. Remember, those are the tiny molecules that literally plug in to other cellular molecules to provide energy to do work. They plug into proteins, for example, the stapler-like molecules or the staple remover-like molecules that allows them to clip molecules together or take them apart. Mitochondria are by far the most abundant organelles in cells. They do mechanical work such as in muscles and where steady supplies of oxygen are needed. You can imagine how many mitochondria you need to produce those energy-rich molecules that are going to be used to drive the muscles, to let you do work. It's curious, mitochondria have one property that has led to interesting speculations about their evolution. Mitochondria have their own separate supplies of genetic material, of DNA. So mitochondria divide like individual little cells on their own. In every cell in your body, the mitochondria themselves are constantly multiplying and dividing on their own to produce more energy centers, more ATP-producing molecules. This also leads to an interesting genetic curiosity. It turns out that the tail portion of the male sperm is discarded during fertilization. And that tail portion is the only part of the male sperm that contains mitochondria, because you're trying to get the tail to move back and forth, to oscillate back and forth as the sperm swims upstream. As a result, when the tail breaks off, the male sperm has no more mitochondria, and the fertilized egg, therefore, has mitochondria only from the female. So every mitochondrion in your body is identical to that of your mother, and her mother, and her mother, and her mother before that, in a maternal lineage. So mitochondrial DNA is an excellent way of tracing maternal lineage. There's no paternal DNA in any of the mitochondria in your body. Another organelle that's in many plants is the chloroplasts. These are organelles that help convert the energy of sunlight into energy-rich molecules. Once again, sugar molecules like glucose, which then can be burned by mitochondria to produce the ATP. Okay, lots of different organelles. So I want to stop for a minute and think about a metaphor that applies to cells that may make this a little bit clearer of how cells work. The nature of cells is much easier to grasp if you think about them as chemical factories, the chemical factories of life. Cells exist to gather raw materials together, to get energy, and then to make copies of themselves in a kind of manufacturing system. And this requires doing chemical operations. So, like every factory, the cell has certain key structures. The cell membranes represent the walls of the factory. The receptors in the cell walls represent the doors, the loading docks, if you will, of that factory. The cell nucleus is like a main office where all the blueprints are stored, where the maintenance records are kept, where the Xerox machine for making more copies of the blueprints exists. You have the endoplasmic reticulum and the Golgi apparatus. These are like the machines that actually manufacture stuff in the middle of your assembly plant, your chemical factory. You have lysosomes, which are like incinerators and shredders that tear up the unwanted material. You have cytoskeleton, which is like the conveyor belts that connect the different part of the factory. You have mitochondria and chloroplasts. Those are like the little power plants that may be used to power the machinery in your factory. In fact, the parallel of mitochondria and a power plant is really quite close. You use sugar in mitochondria to produce energy, and you basically burn a carbohydrate. In most power plants now we use, for example, coal-burning power plants, where you actually burn the coal once again to CO2 and you produce energy. Very analogous. The complex structure of eukaryotes, which have an external membrane and then lots of organelles, each of which has their own membranes, has led University of Amherst biologist Lynn Margulis to a fascinating theory about the origins of eukaryotes. She suggests that eukaryotes evolved more than 2 billion years ago, when relatively large prokaryotes sort of ate and incorporated smaller prokaryotes, large cells containing lots of small cells inside. And then those small cells began to specialize. There are a number of reasons why this might be true. For example, mitochondria and chloroplasts also have their own DNA. They look for all the world like tiny prokaryotes inside eukaryotes. There are also other chemical similarities. Chloroplasts have several structural similarities, for example, to photosynthetic bacteria, which are assumed to be very ancient organisms. Also, the enzymes that make ATP are almost identical in chloroplasts and also in photosynthetic bacteria. But those enzymes differ remarkably from the enzymes that make ATP and other kinds of molecules. So there are all kinds of similarities chemically and physically that suggest that at some ancient point, a large single-celled organism incorporated lots of smaller ones. And that's how eukaryotes came to be. Up to this point, we've talked about two kinds of cells, the prokaryotes and the eukaryotes. But it's possible to group cells in lots of other ways, because cells are remarkable not only for their complexity, but also their ability to specialize in higher animals and in plants. Adult humans have tens of trillions, perhaps as many as 100 trillion cells, and there's several hundred different kinds of cells in our bodies. Each of these cells performs a specific kind of function, a chemical function or a physical function. Each of these cells produces a different suite of enzymes, which is characteristic to that particular function. Under most circumstances, bacterial cells all look and function alike. But multicellular animals are different. They require these specialized structures. Indeed, that's what it is to be a multicellular organism, is to have specialized structures that allow you to function. You have a much larger volume, so therefore you have to have internal structures and external structures, for example. So let's look at a few of these distinctive kinds of cells. We have nerve cells, or neurons. They act as wires in the body. They convey electrical impulses from one part of the body to another. Your body contains more than 100 billion neurons, each of which has several important features. It has an array of fibers called dendrites, which receive incoming information from a variety of points. Then it has a cell body that processes the signal, and it has a long body called an axon. That's essentially like a long wire, and that wire has an insulated sheath that covers it, just like a wire would have. So here the nerve cell serves the function of a wire connecting 2 points in the body. A second kind of cell forms muscles. Muscle cells are amazing. They're composed of many elongated cells that are basically what you call muscle fiber. As much as 90% of that muscle mass comes from two different proteins called actin and myosin. And these elongated proteins can slide over each other, which causes muscle contractions. That contraction is an amazing event. It occurs like a lever process. When you plug in an ATP molecule, it causes a change in shape in the lever and causes two long molecules to slide over each other, which is basically the contraction which occurs when you contract your muscle. Blood. Blood features several different kinds of cells. You have red blood cells, which transport oxygen from the lungs to all the other cells in the body. And these are the most simple, and they're also the most abundant cells in the body. In fact, they've abandoned some of the internal structures that are common to eukaryotic cells. They don't have a nucleus, for example. They don't have their own mitochondria. But they then behave as little more than containers for oxygen that move around the body. But there are incredible numbers of these cells. You're producing new blood cells in your body at the staggering rate of 2.5 million cells per second in a healthy human. And then we have white blood cells, very different kind of blood cells that come in several different types. They combat infections, they consume cellular debris if there's sort of leftover junk in your bloodstream, for example. They also produce antibodies that attack foreign invaders. There are many different kinds of specialized cells that are part of the endocrine system. This regulates bodily functions through the action of chemicals called hormones. So, for example, cells in the pancreas, the thyroid, and other glands manufacture these specialized chemicals that travel throughout the bloodstream, and then they turn on and off other cellular functions, making other chemicals. So we see a huge variety of cells in the human body. Let me summarize this introductory lecture on cells. Cells are the fundamental building blocks of life. They were discovered and studied in ever greater detail using a microscope. And these studies led to three tenets of the cell theory. First of all organisms are composed of cells. Second, cells are the fundamental building block of life. And third, all cells arise from previous cells. There are two main kinds of cells, the prokaryotes, which are single-celled organisms without nucleus, and the eukaryotes, which include both single-celled organisms and the higher plants, animals, and fungi, the multicelled organisms, which contain a nucleus in a variety of internal structures, the organelles. In multicellular organisms, the cells differentiate to serve several functions: nerves and muscles, skin, blood, and so forth. The most remarkable aspect of the cellular differentiation is that every multicellular organism begins as a single cell, as a fertilized egg. Every cell then contains a complete set of genetic instructions, yet each cell produces only the specific enzymes that it needs for its own function. So if one's going to adopt the reductionist approach to life, there has to be something more fundamental, something even smaller than the cell, something that controls life's chemistry. So in the next lecture, we're going to begin our study of the genetic code of life.